and welcome to the Engineering Biology Research Consortium Education Module on Synthetic Biology and Machine Learning. I'm Mary Dunlop. I'm a professor of biomedical engineering at Boston University. And in this series, we are talking with researchers who work at the interface of synthetic biology and machine learning about projects in their group and ideas for how to learn more about this area. Today we're joined by Phil Romero, a professor of biochemistry and chemical and biological engineering at the University of Wisconsin-Madison. Welcome, Phil.
Thanks.
So in this discussion, we're going to be talking about a paper that you published on learning protein sequence function relationships from deep mutational scanning data. And the study compares several different types of machine learning models that range from linear regression to graph convolutional neural networks to even unsupervised prediction methods. This paper was led by Sam Gilman and Phil Romero, and then Anthony Gitter co-supervised this project. It was published in 2021 in the Proceedings of the National Academy of the Sciences. So we'll get into the machine learning parts of the work in a little bit, but can you start by telling us about the overall goal of the study?
Yes. Yeah, so kind of to give some background on this area, my group, we work on protein engineering. And so we're really interested in how a protein's amino acid sequence encodes function. And so when we say function, it can really be any property of a protein sequence. It could be its specificity for binding a particular partner, possibly an enzyme's catalytic activity, its thermodynamic stability. Really, any property of a protein is what we refer to as function. And so if we could understand how sequence encodes function, we would better understand natural evolution. We would be able to predict genetic diseases before they manifest symptoms. And we would also be able to design new proteins with non-natural functions. That's really the goal of my lab, is trying to understand this kind of functional encoding so that we can then make new proteins that do something useful. And so this functional encoding is highly complex. And so we know at a very basic level that a protein's amino acid sequence maps to its three-dimensional structure. It determines how it folds. And this three-dimensional structure positions key functional groups to perform things like catalysis or binding. And so there's this mapping from sequence to structure to function, and understanding this mapping has been like a primary goal of protein biochemistry for the last century. But this mapping also involves, dynamic conformational changes, a practically infinite number of conformational states, poorly understood reaction and functional mechanisms. And so attempts to try to model and simulate this mapping from sequence to function, taking a physics-based approach, have been tremendously difficult, just due to this kind of sheer complexity of thousands of atoms and dynamics and kind of our lack of understanding, right? And so what we're trying to do in this study is, rather than taking this kind of bottom-up approach, trying to model this mapping from sequence to function using kind of physical principles, we're trying to kind of flip it over and do a top-down approach where we just learn this mapping directly from sequence to function from experimental data.
Right. Why is this task particularly well suited to machine learning? What kinds of data do you have for it, and how much of it is there?
Yep. Yeah, so protein engineers and evolutionary biologists like to talk about this concept of a protein fitness landscape. Mm-hmm. And so this is really the kind of an abstract concept where you think about the mapping from sequence to function as forming this very high dimensional surface, mm-hmm. protein sequence space. So we can kind of visualize these in this 2D representation here, where sequences are organized in the XY plane. Adjacent sequences might be differing by a single amino acid substitution, and you can arrange all sequences in this kind of lattice. And then each of those sequences then maps to some fitness value. And here fitness is any property of interest. It could be its biological fitness, but it could be any other property that we might want to engineer. And so we kind of abstract away all the details and we just think about this high dimensional surface, right? And so for machine learning, we basically want to understand what this surface looks like given experimental examples. Mm-hmm. And so this is a really kind of just a classic supervised machine learning approach where we're given sequences and some function value. Mm-hmm. So we have sequences, which are inputs, and each sequence is different. In this slide here, I have kind of colored amino acids showing where they're different. And so we have sequences, and then we want to know how they map to this function value of interest, which we refer to as Y. And so if you're given a data set where you have, tens, hundreds, thousands of these sequence function examples, we then want to try to infer what the overall landscape looks like from this kind of sparse sampling of data.
Right. Yeah, and the supervised part here, just to be super clear, is that you have both the sequence and the function number associated with it. So it's not like you're just given a bunch of sequences and you have to figure things out from that.
Yeah, so supervised learning is where you are given basically input-output mappings, and you try to learn that relationship. And it's probably the most kind of classical machine learning technique, and I think it's also probably one of the easier ones because the inputs and outputs are very well defined. Right. So you have to learn this kind of function that maps between them.
Yeah.
And data, right? Mm-hmm. And so we all know that machine learning methods require lots of data. And protein engineering data can take many different forms. A lot of protein engineering data is what I would consider low throughput, where we're, characterizing protein variants in 96-well plates. We might then perform DNA sequencing on a select few, and we might be able to get data sets that have, tens to hundreds of these sequence function examples. In this particular paper, we were focusing on a semi-recent method. This came out about 10 years ago called deep mutational scanning. And the idea behind deep mutational scanning is that you can combine high throughput screening and selections with next generation sequencing to get these really massive sequence function data sets or proteins or biomolecules or, any biological entity. Mm-hmm. And so how these experiments work is you start off with some gene library that has some randomization to it. And so we will often kind of randomly pick a gene of interest, randomly introduce mutations into it. It might have, single amino acid substitutions, doubles, triples, some kind of distribution of random mutations. We can then take that library and evaluate the corresponding protein activity of each of these variants using high throughput selection methods or screening methods. And so you might be able to, for example, take a protein that confers antibiotic resistance, like a beta-lactamase, put that into a bunch of cells, apply the antibiotic, and for functional variants. My lab, we do a lot of microfluidic screening, so we might be able to put these gene variants into microfluidic droplets and assess enzyme activity in very high throughput. You could use methods like FACS, for example, to measure fluorescence of cells. So any of these high throughput methods, we want to kind of bin these random variants into different functional classes. And so you can take things that are functional or non-functional. You could have high, medium, low activity. There's a lot of ways you can do these experiments, but you really need to just separate these random variants based on the function of interest. And then you just take these different kind of pools of variants with different activity, and then you just throw those on a next generation sequencer. You can use Illumina or PacBio, and that gives you the corresponding genes within these pools, which you can translate to determine the protein sequences. And so in the end, you get these kind of a huge list of sequences that are functional, a huge list of sequences that are non-functional. And from this, you can kind of figure out which parts of the protein are important for determining function versus non-function. And the throughput of these methods with the high throughput screening and NGS, you can get functional data for thousands to millions of protein sequences.
Okay. Yeah, I was going to ask a little bit about the sense of scale here. Can you give a general idea of how much data you're working with?
Yes, I think I will actually get to that
Okay.
In a few slides, but I will say most of these data sets, they're often working with on the order of 100,000 to 1 million protein variants.
Okay.
And putting these types of experiments in the context of this kind of protein fitness landscape that we talked about, this is a very local view of the landscape. Starting from a single point on this landscape, and we're exploring very locally. We're making single and double mutations and just looking very locally. And that doesn't necessarily tell us about the broader landscape structure, but it's very locally what's going on with this protein.
Yeah, that's an important point for thinking about, as you guys show later on, the ability to synthesize novel sequences. You potentially want to look very far out of that space and could run into problems there. All right, so can you explain what the inputs and outputs are for the model and how you represent sequence and function data numerically so that you can use it as input to a model?
Yep. Yeah, so basically the inputs and outputs is just like we have here, where we have an amino acid sequence and we have some function or fitness that was measured from this deep mutational scanning experiment. And so we have this XY pairing, and we have hundreds of thousands to millions of examples of this. Right? And so you can't just plug in an amino acid sequence into kind of a machine learning method. You need to convert this string of amino acid letters, A, C, G, so forth, into a numerical vector that can then be fed into machine learning, right? And so the kind of most traditional way of doing this is called one-hot encoding. And I wish I could actually draw it out here on the screen, but I think that's not going to be possible. But the idea here, it's kind of depicted in this little figure here in the middle where it says one hot, is that you get a string or a sequence of bits. And so there are 20 amino acids. And so you can use 20 bits to represent those. And so alanine is the first amino acid. And so you would represent alanine with a 1 and 19 zeros, right? You would represent, I think, arginine here is the second one, R. You would represent that with a 0, a 1, and then 18 trailing zeros. And so every one of the 20 amino acids has a 1 at a specific position, and the rest of the positions are zeros. And that's called one-hot encoding. And so that's a way of taking these discrete inputs of amino acids and turning them into a vector. And one-hot encoded variables, these categorical variables, can feed very nicely into various kind of analysis, statistical analysis, and machine learning methods.
So if your protein has, say, 300 amino acids, then this would be like a 300 by 20 matrix. Is that correct?
Yes. Okay. Yeah, exactly. But we will often actually flatten that out. So it's actually 300 times 20 in a vector.
Okay.
So that's like the most common way of just representing categorical amino acids and putting them into a vector form. In this particular paper, we also used this kind of physiochemical amino acid featurization. We took these, there's this database called A-index that has all these physiochemical properties of various amino acids, related to things like how large the amino acid is, what its polarity is, what its charge is, and it's a very large kind of vector representation of each amino acid. We compress that using principal component analysis to get, I think it was actually 20-dimensional A index representation as well.
Okay.
And so this A index representation of the amino acid is meant to capture things like the physiochemical properties, things like size, charge, polarity, hydrophobicity, things like that.
And do those just get appended onto the end of the one hot encoding sequence? So you have in the previous example, just for purposes of illustration, it would be 300 times 20, and then you'd start adding numbers after that represent the physiochemical properties.
Yeah, I think it was maybe the first residue had A index 20 variables plus 1 hot, 20 variables, and the second residue had A index plus 1 hot. So in the end, for a protein of length L, I think we have 40 L features, right? So that's the input. And so these are all numbers, and so they can just be fed into various regression methods. And then the output here is just a single number, and that's the DMS fitness score.
And the DMS is?
Sorry, DMS is this deep mutational scanning. DMS. And so, so we basically have sequence to fitness, and then we convert the sequence to this vector representation, and then we train directly on that.
All right, so one of the things that I really like about the study is that you do a really systematic job of comparing different machine learning models. Can you talk about the different approaches you compared?
Yes. Yeah. So first off, I just want to briefly mention, what deep learning is. And so deep learning was this really revolutionary concept that really started gaining traction about 10 years ago. And this paper here is called AlexNet. It's amazing. This paper has almost 110,000 citations in 10 years. And so deep learning was this idea that, neural networks have been around for many decades, but you could basically take neural networks basically pass an input through these various sequential nonlinear activation functions. And so with deep learning, they found that they could stack many, many of these layers, I think something like 20 to 30 of these on top of each other, and that you could get kind of state-of-the-art performance by doing this. And this was kind of going against the more statistical treatment of machine learning. Because these models have many, many more parameters than they have data training examples, but they performed very well for basically any of these very advanced machine learning tasks. And how people usually think about deep learning, where you have these many different layers, is that the kind of earlier layers that are directly acting on the inputs are acting as feature learning. So they're trying to kind of take this raw input, in this figure here, it's an image, and they're trying to extract features from it. And so in an image, you might be extracting things like edges, distinguishing foreground from background, things like that. And then once these earlier layers have learned to kind of look at different features of the input, then the later layers try to process those features of this image, and then try to, for example, classify it or do regression on it to try to map it to a specific value. And so with deep learning, they found that these kind of feature learning steps can really extract very meaningful high-level features from these raw inputs. And they show very good predictive ability when they're applied to new examples that were not in training set. And so in this study, we were wanting to take these same concepts and try to apply them to learning this mapping from sequence to function. And so we wanted to test various model architectures to see how different features of these models affected our ability to learn this mapping. And in machine learning speak, this is often referred to as the inductive bias of a model. So certain models make assumptions about the function that you're trying to learn. And sometimes those assumptions are very well suited for the problem, and the model will actually work better. And so in this study, we compared 4 different types of machine learning models. We performed basic linear regression. And this is kind of a baseline for assessing how well a simple supervised learning model will perform. And so linear regression, as depicted here, really learns the mapping from an amino acid, particular amino acid within the sequence, to function directly. And so this doesn't allow for interactions between sites. It basically treats each site in the protein independently and says, histidine at position 34 might contribute 5 to the score of interest. And it does that for every single amino acid at every single position. And so this is kind of the simplest supervised baseline. People have actually known for a long time that linear regression works fairly well for modeling the sequence.
Yeah, so with this kind of approach, like you're taking each of those individual contributions, weighting them appropriately, and then just kind of summing them together to get your answer. Yeah.
And so then the next step up is a slightly more sophisticated neural network, which we refer to as fully connected. This is also sometimes referred to as like a multi-layer perceptron, MLP. And in these cases, you basically have your sequence as an input, but then you have multiple intermediate layers. Here we're just showing one. And each of these layers has nonlinear activation functions. So what that means is that this, for example, this first white node here in this network is feeding all this linear information from the sequence, but then it's passing it through a nonlinear function. And that basically makes it either on or off. When you have this kind of nonlinearity with many different nodes across many different layers, you can start to learn highly nonlinear relationships between sequence and function.
Can you just give an example to clarify? Like, what would be an example of a nonlinear relationship?
Yeah, so a lot of protein engineers and evolutionary biologists talk about this concept of epistasis. So that's where 2 mutations in a protein interact in some way to give a kind of behavior that is non-additive.
Yeah.
And so the simplest example could be if you have 2 residues, say an arginine and an aspartic acid that are forming a salt bridge, you have a positive and negative charge, they're interacting with each other happily, right? If you mutate the aspartic acid to an arginine, then you have an arginine and arginine that are very close together, and you get this charge repulsion, so that's favorable. But then if you mutate the original arginine to aspartic acid, you essentially swap the polarity, you have a salt bridge, right? And so that's an example of how you can have interactions between residues, right?
Okay, so that cannot be captured with a linear regression, but these more complex networks that have the nonlinearities and incorporating them can. Yeah.
The linear regression treats everything independently. So it will just say arginine at this position is good, aspartic acid at this position is good. But when you change it together, you can actually get these clashes. And the only way to represent these interactions between residues is to use a nonlinear model. So this fully connected network can represent that, and it can learn it from the data. And then kind of the next level up we use these convolutional neural networks. And so convolutional neural networks are widely used in image processing and computer vision.
Mm-hmm.
And so they're actually depicted here. It's where you have these kind of little windows, which are called convolutional filters, that scan over the image. And the filters have their own weights, and they can learn to identify aspects of the image, for example, like edges.
Right.
If it learns to detect edges, everywhere it sees an edge in this image, it will light up. You can then pass it through these multiple convolutional layers to get these really kind of complex representation and learning of features. And so our first convolutional model, we are applying this same kind of convolutional scanning approach, but directly to the amino acid sequence. And so we have a little window, and we can scan across the sequence, and we try to learn convolutional filters that will find patterns in the primary amino acid sequence of the protein.
So with the image analysis, you talked about edge detection as a potential feature you could pick up. Could you give like an example with protein sequences that you might?
Yeah, so protein sequences also have some kind of signatures you might want to look for. Maybe one of the examples that comes to mind first is often if you have a beta strand in a protein, the residues will alternate. So like the even residues will be on one face and the odd residues will be on the other face of the beta strand. And so if this beta strand is pointing to the core in one direction and is solvent exposed in the other direction, you will get this alternating pattern of hydrophobic and hydrophilic amino acids alternating back and forth. And so a convolutional filter will be able to identify those types of patterns from an amino acid sequence.
Yeah.
And possibly if it learns that you should have this alternating pattern protein, and then you make a mutation that breaks that pattern, you'll be able to say this pattern is broken here, and it's not broken in kind of the good sequences, and so maybe this is a bad protein. Right?
Okay.
And an important aspect of convolutions is that they are called parameter sharing models. And so this convolutional filter, the same convolutional filters are applied across the entire protein sequence. So in the previous model, the fully connected model, it kind of learns specific aspects of the input. So it learns exactly like residue 5, tyrosine is a good residue. But parameter sharing models are actually learning general things across the entire amino acid sequence. And so we thought that this might be able to start to learn these more general rules of protein biophysics rather than specific, like this protein family likes residue 5 to be tyrosine. Does that make sense? Yeah, so that's the sequence convolution where we're applying this kind of sliding window across the amino acid sequence. The next level up, we applied what is called a graph convolutional neural network. And so here we actually represented the protein's three-dimensional structure as a graph. So if you look at a protein, its sequence folds up into this three-dimensional shape. Sometimes, the residues at the beginning and the end can actually become close together in three-dimensional space, so it folds up. And so what we do is we go to every residue in the protein and we say, what other residues are within, I think, actually we varied this distance, but it was like, say, within 7 angstroms of this residue. And so if you look at residue 5, maybe it's within 7 angstroms of residue 4 and 6, but also maybe residue like 300, right? And so this defines a graph structure that represents which residues are close together in three-dimensional space. You can then apply these same convolutional methods, but to the graph rather than the sequence.
Okay.
You kind of look at the local sequence information, local graph, and say what is nearby, and try to learn graph features. And so just like we might be able to identify sequence patterns of alternating residues for a beta strand, looking at the structure graph, we might be able to learn more general structural features, like whether a residue is in a protein core or whether it's on the surface, it's solvent exposed. So this method is meant to incorporate structural information into these neural networks. And so we define these neural network architectures, right? These kind of basic models. And then what the graduate student Sam Gilman did is he scanned through tons of different options where he's looking at, how many layers each of these models has, different numbers of filters for the convolutional models, kernel sizes, that's like how big of the sequence window it's looking at. Graph distance threshold is the angstroms, so our neighbor is 4 angstroms apart or 10 angstroms apart. And then in the training process, you also need to look at things like batch size, so how many examples are fed into the gradient descent algorithm sequentially, and then the learning rate is kind of how far you jump when you update the model. So this is a kind of a fairly standard approach in machine learning where you kind of come up with the models you want, and then you just try a lot of different options, and you do your kind of training, validation, and testing, and take the model that has the best performance.
Actually, can you talk just really briefly about the training, validation, testing approach? How did you, when you went through and tested these different combinations of hyperparameters, how were you systematically evaluating things?
Something like, say we had a data set with 1 million sequences. I think we split that up into 60% is training, 20% is validation, 20% is testing. And I think when we're making these decisions here for which hyperparameter combination is best, we actually only look at validation. And so we're basically training on training data, testing the performance on the validation data. Once we've finalized the modeling decision, then we test its performance on the test data, because we don't want to influence the model in any way based on the test data.
Right. Okay.
In general, in machine learning, you have to be very careful about these things, how much information it's getting through. And there's a lot of ways you can kind of trick yourself into thinking a model is performing well. You let information from your test set kind of influence your training process.
Can you talk a little bit about how the models perform? Are there any sort of specific takeaways that you'd like to highlight from the results?
Yep. So in this paper, we had 5 very diverse deep mutational scanning data sets. These are completely different proteins with different kind of functions. We also had very different sequence lengths and data set sizes. In these plots here, we show our supervised learning methods at the very bottom. That's linear regression, fully connected, convolutional neural network, and graph convolutional network. We first actually compared these to existing prediction methods for proteins. And so the first one at the very top is called Rosetta. This is a kind of structure-based physical modeling method. And so in comparison, you see that these supervised models are performing much better than this. This is not surprising because the supervised models they're trained on the data, and we're asking it to predict this same function, whereas Rosetta is probably capturing something more related to the protein's folding and structural stability, right? Like it doesn't know anything about the specific function of the model. We also compared it to these unsupervised learning methods that are widely used for protein prediction. One is called EV mutation, and one is called deep sequence. And so these methods are trained on large multiple sequence alignments of a protein family. And so you could, for example, get a multiple sequence alignment of GFP where you do a blast search. For example, you might get hundreds of thousands of GFP homologs. These are things that are evolutionarily related to GFP. And then it tries to perform unsupervised learning. So it doesn't have a label. It just has this big list of sequences, and it tries to learn what mutations are good or bad in GFP from this unlabeled data, right? And that's a very hard prediction task too, because we're not giving it any labels. And so it's not surprising that these supervised models that we're training are significantly outperforming unsupervised models. Right. And so supervised models outperform these other methods. That's pretty obvious. We also really see that the performance varies drastically across the proteins. So, for example, this small protein GB1, basically every model is capping out at very close to a Pearson's R of 1, which is perfect prediction, whereas this BGL3 beta-glucosidase enzyme, the performance is much more modest, something on the order of 0.5 Pearson's R, right? And really the performance of these models is a very complex function of kind of how big the protein is versus how many data points you have. So for BGL3, it's 500 amino acids long, and we only have 26,000 data points, versus GB1, it's 56 amino acids long, and we have over 500,000 data points. And then there's also a data quality aspect to it as well. Some data sets, maybe the high throughput screen is less reliable, and therefore you might get more false positives, and data in general is just low quality. So this is kind of how the performance is on full data sets. One thing that's really informative to look at for these machine learning models is how they perform on smaller data sets. So basically, dialing down the training set size. So say only giving the model 100 examples to train on, 1,000 examples to train on, and kind of scanning this entire range of different training set sizes and assessing them. And this is where we really start to see a large difference between the convolutional models and the kind of more simple linear regression and fully connected models. And so I think this is probably a little small to see on the screen, but GFP, for example, the parameter sharing models are learning much more quickly from small data, the fully connected or linear regression model. And so if you look at performance at say 100 sequences, that's almost as good as linear regression trained on 100,000 sequences, right?
And what would you consider like a pretty typical training set size that would be accessible for this type of data?
Yeah, so I mean, this is a huge challenge, an important problem for the field is, sometimes we have access to high throughput methods like deep mutational scanning, and we can get, on the order of 100,000 to a million sequences. But oftentimes we are kind of stuck with much lower throughput methods just because the protein of interest can't be characterized in high throughput.
Yeah.
So we have to use things like GCMS or 96-well plate screening. In those cases, our data sets are really limited to something like, on the order of 100 to 1,000 examples. And so that is a problem that a lot of people, including us, are working on in the field, is trying to make more data efficient deep learning models.
Yeah.
So taking the power of these deep learning approaches, but then trying to get them to work in this low data regime.
Yeah, which is often encountered in synthetic biology. Yeah.
Yeah. So I put that in red at the bottom, is that all of these models require at least thousands of training examples to get high performance. And so that's a major limitation of the approach.
Yeah.
Another thing that we found that was very interesting is that the structural information in the graph convolutional network really did not seem to improve the performance at all across anything we tested.
Yeah, interesting.
It's providing more information, and the protein structure should be more directly linked to function than just the sequence. Mm-hmm. And we have a number of hypotheses about this, possibly that these data sets are so local in sequence space that it's maybe locally like linear or well behaved. Maybe if you modeled further out, then structural information would be important. Another thing we had some evidence for is that the kind of just the complexity of these neural networks, there's so much complexity to it that it's still able to learn this kind of structural information embedded in the graph convolution, even if we don't give it structural information. So that was a very interesting observation that we did.
Can you talk a little bit about where the data come from? Because I think some people in the synthetic biology field who are interested in getting into this aren't in a position where they themselves can generate incredibly large data sets. These 5 different diverse deep mutational scanning data sets, are these things that you guys generated in your lab, or were you taking data from the literature? Can you talk a little bit more about that?
Yeah. So there are quite a few data sets out there. So I actually generated this BGL3 data set during my postdoc. My lab has actually generated quite a few other deep mutational scanning data sets. But there are a lot of data sets that are publicly available as well. So the other 4 data sets are publicly available. I think there's probably about 30 deep mutational scanning data sets that people will regularly analyze and use as benchmarks for these machine learning methods.
Nice.
Yeah. And there are even some databases out there that have protein sequence function data. For example, there's one called Protobank that tries to store lots of sequence function data that you can kind of program API with to download and analyze it all automatically.
Machine learning models, especially the more complex ones, are sometimes criticized as being black boxes that don't provide insight into structure or mechanism. But you showed that your models are interpretable. Can you explain how you do this and what you found?
Yep. Yeah. So it is true that there's a lot going on inside these neural networks, and it is very hard to interpret what exactly they are learning. And so in this paper, we applied a number of kind of standard explainable AI interpretation methods. One of them, which I'll show you here, this video crashes if I play it in slideshow, so I'm just going to play it here, is that you can basically visualize the relationships between different amino acid sequences in these data sets. This is GB1. As it passes this information from sequence to this last layer. And so we're using a method called UMAP that kind of tries to do a 2-dimensional representation of these possibly high-dimensional spaces. And so we can look at how the relationships between these sequences changes. And we had the input. We saw that the red and blue sequences were very kind of mixed together. That means low fitness sequences and high fitness sequences were very close together. But as you pass it through this network, it kind of arranges it such that high fitness sequences are on one end and low fitness sequences are on the other end. And we actually looked at this a little closer. And what we saw is that there were these kind of 3 different clusters of inactive sequences. And so we have one way of being high fitness, and that's red. There's kind of 3 different ways of being low fitness.
Okay.
And so we looked a little closer at what mutations were present in these different clusters. And what we found was really interesting to us was that it seemed like the mutations were occurring in different parts of the protein. And so this very far left cluster, which is circled in blue, tended to be residues that were directly on the binding interface for this protein. Mm-hmm. So this is called GB1. It binds to IgG. So they're on the binding interface, whereas the other 2 were on specific beta strands that were kind of packing into the core of the protein. And this is still very hypothetical at this point, but we're thinking that maybe it was able to learn different ways that the protein is becoming inactivated. Mm-hmm. As you could inactivate a protein by disrupting the binding interface, you could also inactivate a protein by mutating core residues that cause the entire thing to unfold.
Yeah. Uh-huh.
And so we still have to explore this a lot. This is conjecture, but it would be really amazing if these models could start to dissect mechanisms of protein inactivation from this type of data.
Yeah, yeah.
Another way that we can kind of do model interpretation, did some integrated gradients, feature importance. So this basically tells you which features of the protein are most informative, are most important for this functional output. We can map this onto the three-dimensional structure of the protein, and we see that these kind of blue residues here are directly involved with this binding interaction. This is PAB1. It's a poly-A binding protein, and so it's directly involved in this binding interaction, and the feature importance identifies these very key residues for this. I think probably the easiest way to use these models to understand the protein, though, is to kind of just think about it as a kind of sequence function black box. And so at the bottom here, we basically just took this trained model and then we made all single amino acid substitutions and evaluated it with the model. So even if these amino acid substitutions weren't present in the data, we still get a kind of a predicted value.
Yeah. Okay.
And so I think this is a very kind of straightforward and easy way. It's not using necessarily these fancy explainable AI methods. It's just using it as a sequence function model and trying to understand the protein that way.
Yeah, so you're effectively getting like a lookup table out of like, okay, if I wanted to know what this combination and that worked out too. Yeah. Okay, so of course, one of the things that synthetic biologists are often in protein designs, and you took some steps in that direction with this manuscript. Can you talk a little bit about how you did this and how it worked in practice?
Yep. Yeah. So here the goal is to basically the deep mutational scanning really provides this dense local sampling of the landscape. So we're looking at single and double mutations within a given protein. And for protein design, we're really wanting to try to move further away because we've already experimentally characterized this kind of local space. And so one of the questions we wanted to ask is, can we extrapolate these models, right? And a lot of people in machine learning tell you should not be doing extrapolation away from your training. But it's really important for us in protein design to be able to move further away and predict things that are better than anything that we saw experimentally. And so the scenario is basically this here, where we have this local sampling and we're trying to predict away. And the protein, just very quickly, is this very small protein, GB1. It binds to IgG antibodies. It's very small. Our models on this protein were extremely high accuracy due to this massive deep mutational scanning data set. And so we use these neural network models to kind of search sequence space, trying to predict these highly fit designs. And we actually designed 5 sequences with varying distance from this wild type. So if wild type is at some point in the landscape, we designed a sequence that was 10 mutations away, 20 mutations away, 30 mutations away, 40, and then 50. And these are kind of radii in this fitness landscape moving out, right? And then we went into the lab and we tested these. We found that nearly all of them were dead. Design 20, 30, 40, and 50. They did not express the soluble proteins, but the design 10, which had 10 mutations from wild type, actually did express as a soluble protein, which was very exciting to us. This is only 56 residues long, and so we're changing on the order of 15 to 20% of the amino acid sequence of this protein. And so, we went to the lab, we started testing it, we did circular dichroism spectroscopy to show that it has similar secondary structure content to wild type. Here we predicted its three-dimensional structure using Rosetta. It was nearly superimposable. And so these suggest that its structure is very similar to wild type GB1. We're actually determining the structure by NMR currently. what was really exciting to us is that it had super high binding affinity for IgG. And so if we do these kind of IgG titrations, in this range, wild type is basically hardly binding at all. These other two variants, A24Y and E19Q A24Y, are the two best single and double mutants from the training data. Those are also hardly binding. And then we look at this design 10 sequence, and it's starting to saturate by 100 nanomolar. And so it's hard to estimate it just based on these curves, but we think it's on the order of a 60-fold increase in binding affinity.
It's fantastic that you got this, even any of these to work at all. When you were initially designing this, did you have like some preconception about what was going to work, or like when you clearly tried, 10, 20, 30, 40, 50, like did you think there was some chance things were going to work very far away, or did you think nothing was going to work at all? Yeah.
We do these types of experiments a lot, and we usually expect them to all fail. Just incorporating 10 mutations into any protein is likely to break it. But yeah, we found over and over again that with these machine learning models, we can introduce 10 mutations, usually no problem,
That's
and they
awesome.
often have improved activity. Yeah, and something that is not in this paper, but now that we're working on, is that we performed this kind of large-scale, high-throughput protein design, where we designed thousands of variants using this model. And now we also have some GB1 designs with, for example, 50 mutations, 50 of 56 residues mutated, that still fold. We really do think that this neural network design is kind of exploiting these nonlinear features of the landscape. Because we see this design 10 is not simply composed of the top 10 single mutations. It's really combining mutations that have synergistic effects with each other. And design 10's predicted fitness is over 2 times greater than the variant with the top 10 single mutations. So it really is kind of finding synergies between residues, nonlinear synergies, and taking advantage of that to find these high fitness designs in the landscape.